Okay, so at the end of 17.3, we made our mature mRNAs. So they were built during transcription in the nucleus, and then they underwent mRNA processing, either the adding of the 5' cap, poly A tail at the 3' end, and any splicing that's going to happen. And now they're ready to be translated. So in transcription, we were kind of rewriting the same message in the same language. DNA and RNA are both nucleotides. They are both nucleic acids, so it's the same language. But now we're going to proteins, to polypeptides and amino acids, so we're at a totally different language now, so we have to translate it. And the translator that we're going to use is a molecule called tRNA. So that's like the basics. So we're going here from mRNA to protein through translation. Like I said, the translator is this molecule called tRNA, which stands for transfer RNA. And that's what we're going to use to turn our mRNA message into a polypeptide. And what they do is the tRNAs read the amino acid or the RNA message, and they bring in amino acids, and then to add to the growing polypeptide chain. And it's simple in theory, but it's actually a very complicated process, and we're going to learn all about it. So here's the basics of translation. So translation happens at the ribosome in the nucleus. So ribosomes are what build polypeptides, proteins. And basically, they read our mRNA message, which we just made during translation. So the mRNA just moves through the ribosome and gets read. It gets read 3 base pairs at a time at our codons. Remember, this is our reading frame, so these 3 are read together, then these 3 are read together, then these 3 are read together. What happens is the tRNA translates this message by having a part that is RNA, so it has a complementary section to the mRNA strand, and at the other end is an amino acid. So for example, this is the next codon that's going to get read, GGC. So tRNA is complementary CCG, and it carries a glycine. And that anticodon right here is going to meet up with the codon, has a glycine attached. It's going to move through the ribosome, and this glycine is going to get added on to this growing polypeptide chain. And it's just going to keep happening over and over again. And the tRNAs leave when they're done. So it is a process where the tRNAs are reading, the ribosomes reading the message and using the tRNA to translate it into a polypeptide. So to properly understand translation, we have to understand the translator, or tRNA. So all of our tRNAs are not the same. And that's important that they're different because we need to be able to translate different messages. Each has an amino acid on one end, like we just saw right here. Here's an amino acid on one end, and on the other end are 3 base pairs of RNA, and that's called our anticodon. So it's like a codon on our mRNA, except it's an anticodon. And it is complementary to a codon on the mRNA strand. So those are the 2 important parts that help us to translate, but they actually are made up of a lot more RNA than just that. They're usually about 80 base pairs of RNA, which is different than an mRNA molecule, which is usually about several hundred base pairs long. So it's a pretty short mRNA or RNA molecule. It's not mRNA, it's just RNA. And it folds up all on itself, which makes sense. So it's single-stranded. RNA is single-stranded, but it likes to make hydrogen bonds with other bases, so it usually will fold up on itself into this 3-dimensional structure. So if it gets flattened onto a paper, which it doesn't in real life, but if we were going to draw it, it looks like this, kind of like a clover. You can see all the base pairs. They form all these loops. And at one end, this is where the amino acid is going to get attached at the 3' end of the tRNA molecule. And down here at the other end are the 3 bases that act as the anticodon. So this is it flattened. This is an attempt at a 3-dimensional structure. And you can see the 3' end up here of the RNA molecule, which is going to be where the amino acid attaches. And down here is the anticodon. And I am covering up this picture, but this is on page 338 in your textbook if you want to look at it. This is what the textbook uses to demonstrate what a tRNA molecule looks like, just for the sake of making things easier when we're modeling translation. So again, the amino acid is attached up here at the 3' end, and down here, which you can't see kind of, are the 3 base pairs of the anticodon. But remember that the rest of this is made up of RNA also. Oh, there it is. There's the anticodon. Okay, so there's two important steps, two important molecular recognitions where molecules need to recognize each other correctly for translation to happen. And the first one is we need to make sure that the correct amino acid is attached to our tRNA. So tRNAs are used over and over and over and over again. So it's not like the amino acid's always on there because it's bringing the amino acid to the polypeptide chain and then it gives it up. So the tRNA needs to be able to be used multiple times for that. And so it's important that we can correctly match the right amino acid onto that tRNA. And there are very specific molecules called amino-acyl tRNA synthetases that do this. Crazy name. Basically what they do is they find the correct tRNA. You don't really need to know the details of this very much. But they find the correct tRNA and the correct amino acid, and they bring them together. That way we have our tRNA that has the amino acid on it. So that's the important first step, is we need the amino-acyl tRNA synthetases to correctly attach the amino acids to the tRNAs. And there are we need a different amino-acyl tRNA synthetase for each of these, for each of the amino acids. So there's 20 different kinds of amino-acyl tRNA synthetases. The second important pairing or molecular recognition is when the tRNA anticodon matches with the RNA or the mRNA codon. This is really important because this is where the translation actually happens. So we need to bring the correct amino acid to the growing polypeptide chain. If we have a code for phenylalanine, we need a phenylalanine to be there. And that's why we have this codon and anticodon system. So basically, do I have a picture? Sorry. Okay, so here's our anticodon, and here's our codon. So we need these to correctly match up. So like this UUU right here is coding for phenylalanine. And we need that UUU. We need a phenylalanine at this point in the growing polypeptide chain. So it's really important that that recognition happens. If you look at the table on page 17.1, on page 330, table 17.5, I don't have it here, it's the amino acid codon table. So it says which codons code for which amino acids. You can see, or you should know, that there's 20 different amino acids, but there's actually 61 different codons because 3 code for stop with no amino acid. So the reason, and let me get this code, let me get this table. Okay. So here is the table. So here are all of the amino acids, or these are the amino acids right here, and here's all the codons. So there's 20 different amino acids, and there's 61 codons because these 3 do not bring an amino acid, but there's only 45 tRNAs, which doesn't make a lot of sense, right? Because there's 45 codons, so there should be, or 61 codons, there should be 61 tRNAs. But it turns out that the third base pair, or the third base, so with these guys right here, the UCAG right here, UCAG, UCAG, the very third ones, don't matter as much. For example, if the codon reads AAA and the anticodon reads UUC, it may still, they don't match up. It should be a U to match this, but it would still bring the proper amino acid. I wish that there were a picture for this, but it's called the wobble hypothesis. Basically, what it means is that that third base isn't as important. So the wobble hypothesis says that as long as the first 2 bases match perfectly, then sometimes there's flexibility in the third base, which is why there's only 45 tRNAs with 61 codons for amino acids. And it's also why, if you look at that table, shouldn't have gotten rid of it. If you look at that table, and you can see that, for example, all of these code for glycine, and that's because their first 2 are the same. Whereas over here, like GGA codes for glycine, GAA codes for glutamic acid. So it's okay if the third base pair is a little bit different, and that's why when we have multiple codons that code for the same amino acid, they're usually only different in the third base pair. And that is the wobble hypothesis. Okay, that's the wobble hypothesis. Allows some tRNAs to bind to more than one codon, et cetera, et cetera. So ribosomes are where this happens, right? Where this translation actually happens. So they facilitate the binding of the tRNA anticodon with the mRNA codon as we're building our proteins. And it's important that we understand the structure of proteins while we're doing this. So proteins are made of two subunits, a large subunit and a small subunit. And they're made of proteins and ribosomal RNA, which we call rRNA. In eukaryotic cells, these are made in the nucleolus, if you remember the function of the nucleolus. And both bacterial and eukaryotic ribosomes are very similar, but they have a couple of significant differences, and this is why. Those differences are how antibiotics work. So a lot of antibiotic drugs, like if you get sick with a bacterial infection and you take medicine for it, those drugs target the ribosomes of the bacteria because the bacteria will stop making protein and they'll die, but we want to make sure they're not also going to attack our ribosomes. So since a lot of the ribosomes are different between bacteria and eukaryotic cells, that's one way that we use antibiotics to attack bacterial cells. So here is, this picture's on page 339. It's 3 different ways to look at it. So here's like a computer modeled one. Here's a schematic one showing the binding sites and a schematic model with RNA and tRNA. So there's 3 places in the mRNA molecule, or in the ribosome molecule, that we're going to talk about. There's the exit site, the P site, and the A site. And tRNAs enter the ribosome at the A site. At the P site, they attach their amino acid to the growing polypeptide chain, and then at the exit site, they're moving out. And then down here is where the mRNA binds. So the mRNA moves through the bottom, and then the E, P, and A sites are in the large subunit of the ribosome. So the P site holds the tRNA that carries the growing polypeptide chain. So here, the tRNA is attached to the entire polypeptide chain. So it has here its own amino acid that it always carries, and the rest of them for this entire growing polypeptide from the mRNA instructions. That's the P site in the middle. The A site holds a tRNA that carries the next amino acid to be added to the chain. So whatever the next codon reads, that tRNA comes in, and it hasn't started interacting yet, but it will in just a minute. This will move into the P site, and then this will have all of the amino acids on it. And then there's the E site, where the last tRNA to give up its amino acid is leaving. So here, this tRNA was just in the E site, and so this triangle amino acid was the one that belonged to this tRNA. And the tRNA that left before this one would have had this square amino acid attached to it. So the tRNAs move through and they pass on the growing amino acid chain.